Well, welcome everyone. My name is Dr. Mark Israel. I'm the National Executive Director of the Israel Cancer Research Fund. Many of you know that we're a nonprofit organization founded in 1975 by a group of American and Canadian physicians and scientists who recognize the extraordinary accomplishments and unique global impact of science in Israel. And today, the Israel Cancer Research Fund has awarded more than $78 million in funding for over 2,500 grants to hundreds of Israeli scientists, many of whom have conducted truly groundbreaking research. And during this time, we've supported individuals at essentially all of the leading research institutions in Israel, something that enables us to invest directly in the very best scientists and their best ideas each year. The key value added by ICRF is bringing donors and researchers alike to our rigorous grant proposal review process that engages leading physicians and scientists from major research institutions throughout North America. These individuals review each grant application and prioritize them for funding. And some of you know that ICRF scientists have been instrumental in many, many discoveries that are now immersed in the fabric of what we know about cancer. Some of these discoveries have led directly to the development of breakthrough FDA-approved drugs, Gleevec, Doxil, Velcade. Velcade, in fact, is a drug whose very existence rested on the work of two ICRF-funded investigators who went on to win the Nobel Prize in Biochemistry, Dr. Avram Hershko and Dr. Aaron Ciechanover. Further, I'm happy to report that ICRF grantees continue to make major breakthroughs and at their forefront of cancer discoveries in nanomedicine, immunotherapy, stem cell research, targeted therapy, amongst many other areas of cancer medicine. And that we owe great thanks to many of you in this audience who've been strong supporters of our efforts to engage Israeli cancer science. And we value your interest in ICRF immensely. So today it gives me great pleasure to introduce a fellow cancer scientist and friend of almost 40 years, Dr. Howard Cedar. Howard's actually coming to you from a hospital bed in Hadassah Hospital where he landed after a bicycle accident a day or so. But you should know that Howard is now Professor Emeritus in the Department of Developmental Biology and Cancer Research at Hebrew University and is renowned, and I knew him both as a scientist and friend, as being renowned for truly spearheading breakthroughs in the field of DNA methylation and cancer epigenetics. And his findings have had a profound impact on our understanding of tumor biology and disease development of many kinds. Howard was educated, he was born in America, was educated at MIT and earned his MD and PhD degrees from New York University. And amongst a very long list of truly internationally renowned prizes, Howard is the recipient of the Emmett Prize, the Wolf Prize, which he won in conjunction with Professor Rosen, and of the Israel Prize, perhaps the most highly regarded prize in all of Israel for science. And today Howard's going to talk about his groundbreaking research in an area called epigenetics. And then he's going to share how these fundamental breakthroughs in our understandings of how genes work has impacted on cancer. And now, before I turn this over to Howard, let me suggest that if you have questions for him, that you enter them into the chat function and address them to me. The chat function can be found at the bottom of your screen. And once you click on it, it'll be intuitive how to send me a question. And I'll then try to pull together the questions and present them to Howard in a way that we can be parsimonious in bringing together the themes that these questions identify. And so without another moment passing, let me turn this over to Howard. Howard, welcome to this meeting.
Hi, everybody. It's good to be with you today. I'm happy to be able, as you can see, I'm happy to be able to do this and to share with you some the things that I think are really interesting and may represent one of the future directions in dealing with oncology and dealing with tumors. So all of what I'm going to tell you about started with basic research. I'm devoted to basic research. Our laboratory is devoted to basic research. And we did this in the understanding that to really get to the bottom of any problem, whether it be medical or whether it be agricultural, one must understand the underlying mechanisms and understand how cells work, with the hope that someday those will be put into action and be used for something useful, in the case of cancer, either for treatment or, as I'm going to tell you a little bit later, also diagnosis. So our story starts really basically, and hopefully I'm going to make it simple for you. What you see here is DNA. DNA contains our genes. You all know that the genes are written in a language, the language of DNA. Like all languages, it has letters and words and sentences. And what you see here is some information in the DNA in the form of letters. And it's the string of these letters that determines what the information is. And then this information is used to make something that the cell needs. And that sounds very simple. In other words, every time a cell of the body is supposed to do what we want it to do, it runs to its book, the DNA, it looks up what it has to do, and it makes the proper components of the cell to make that cell whatever it is. Okay? But it turns out that, and this was our discovery many, many years ago, that this language of DNA is more complex than we thought. Actually, this language can be annotated. In other words, there's a way of taking this language and marking it in order to help the reader, in this case the cell, understand what's written there. And this marking is in a simple chemical called methylation. And the way it works is that if you have information for something, if it's in the cell, if it's unmethylated, if there's no methyl, then that information is used. However, if it's methylated, the information is not used. So I'll just give you an example here. Here you see three different types of cells. The iris of the eye, you see. These are liver cells, these are nerve cells. They all have exactly the same DNA book. They all have exactly the same information. Now the eye, the iris of the eye, makes a component called blue pigment, in this case, blue. Okay. But it's only made in the eye. The information for this component, for this pigment, is in every cell. And what DNA methylation does, it enables cells to know what's appropriate for that cell. So here's the DNA, here are the genes. The same gene in the eye, in the liver, and in nerve cells. But when we look at the level of methylation, we see that in the liver, it's methylated. Remember what I said? It's methylated, it's not used. And in the nerve cells, it's methylated, it's not used. Only in the case of the eye is that information used. Okay? That's how methylation works. It's called an epigenetic factor as opposed to just a genetic factor. Here's another example. This is a component that's only made in the liver. However, the information, the gene, is in every cell of the body equivalently. But if we look at the epigenetic layer, we see that it's methylated in the eye, methylated in the nerve cells, but unmethylated in the liver. And that's why the liver makes the proper components. Even though it has all the information for making every component in the body, there are these signs, this epigenetic layer that tells it what should be read. Okay? As you can imagine, this is a very, very important part of biology for all sorts of things. And I just want to give you, so you'll be able to translate this into something more understandable, I've taken here another text. This is the text of the Bible. This particular book was written down 1,000 years ago and has been stored in a synagogue in Damascus. It contains the entire Bible, and it looks like, where you see here, it looks like this. When you take apart this information, you see the following. There's text, there are letters, just like in DNA, there are letters. But you know, in Hebrew, just having this text is not enough. This is quite difficult to read, even for a Hebrew speaker. But what this book contains is an annotation. It has the vowels and how the words should be said and where are the ends of the sentences. And that's the epigenetic information. Okay? And that's the way it works in our body. We have information, and the information can be annotated by signs and symbols in order to tell cells what to use and what not to use. This is a very important part of cancer. So I'm sure you all know that tumors begin from normal cells. These are normal cells. They behave nicely. They're all lined up. The cells don't attack each other. They don't grow too much. But when they become transformed into a tumor, they start to grow, first slowly, then more rapidly. And you'll notice as they grow, they also change the color, the shape, the aggressiveness, the ability to attack other tissues. All this happens from this one cell. Now, interestingly, if you look in the normal cell and compare it to the tumor cell, it has the same, almost exactly the same DNA. But what's different is the annotation. Different is the methylation. So when this cell becomes a tumor, the methylation changes. Here, what you see are genes in yellow. Those are methylated. And information that's unmethylated in blue. And when this becomes a tumor, many of the ones that are methylated and were turned not used now start becoming used. And many of the ones that were used now start becoming unused. So because of this change in cancer, it changes the way the cell behaves. And that's what gives us all of the properties of tumors. In early research based on methylation, it was possible to develop a drug. Right? And this drug was tested in animals. And this is looking at a particular tumor in mice. You can see that a large number of tumors are formed in mice. But when you treat them when they're young with a drug that doesn't allow those methylation changes to occur, it prevents the appearance of tumors. And these drugs are being used in people as well for a variety of different tumors and for a variety of different reasons. But now I want to show you something that I think is really amazing. And this is work that came, not basic, it's applied research, but it came out of all of this basic research, and you'll see what the importance is now. So what you see here is you're looking inside a blood vessel. You see blood cells, different kinds. There are red blood cells and white blood cells. But in addition, you see pieces of DNA. DNA is not supposed to be like that. It's supposed to be in the cells, not outside the cells. So where does this DNA come from? It comes from other cells in the body when they die. A cell dies, it loses its information. This information then goes into the bloodstream and circulates in the body. So we've taken advantage of this by developing a simple test to look at this free DNA. Now you could say, well, it's not going to help us very much because the information is the same in every cell. So what difference does it make if I look at the blood DNA, all the DNA is going to be the same. No matter what cell is dying in the body, has the same DNA. But it's not true because every cell has a different pattern of methylation. And we can take advantage of this to see where the dying cells are coming from. And one of the places where many dying cells come from is in tumors. So we can use this test. It's a simple blood test to look at the DNA, look at the information, measure its methylation, and determine what cells it comes from. Which cell? Is this coming from the colon? Is it coming from the breast? Is it coming from the pancreas? Okay. And I'll give you an example of how this works. Take a look here. These are people who are known to have pancreatic cancer at all sorts of different stages, from stage 1 to stage 4. Okay. And what we've done is we've taken blood from these patients, and we've taken blood from people who are not ill. They don't have cancer. Right? And we've taken blood from patients who do have cancer. And we've looked for the methylation to determine what cells the DNA comes from. And you can see all of the patients are positive, and all of the people who are well don't show any pancreas DNA in their blood. Now, this has been expanded to a relatively large test on many people with cancer. So I'm going to go through this with you. This study looked at, as you can see, 6,500 people, 6,500 blood samples, 4,000 from cancer patients, and 2,500 from people who were never diagnosed with cancer. And we did the test. And the test successfully determined which patients have cancer and in what tissue. Okay? Over almost about 85% of the people known to have cancer were diagnosed correctly by this method. Okay? And there were only a few, and the people who are normal, there were only a few, very few. Out of 2,500 normal people, only 1% of them showed positive tests for cancer. So this is very good, but it's not really what we'd like. We'd like to have a test that will determine whether a person has cancer before he has signs of cancer. And here you can see the initial results of such a thing. So these 25 patients, when they were used in the study, these 25 patients who showed tumors, even though they didn't have any tumor, they showed the test was positive, even though they didn't have any tumors. Within 15 months, 10 of these developed tumors. And that's only 15 months. In other words, this test has tremendous potential for picking up cases of cancer and determining where all over the body. Now, I'd just like to show you my wish for the future. This is a blood report that you usually get when you, in Israel, when you go to your medical supplier, right, your HMO, and they take blood tests. You get something like this, okay? They say the different chemicals in the blood, and they tell you whether it's normal or not normal. Well, our wish is that someday we'll be able to take a patient and every 6 months look at his blood, measure the methylation, and be able to tell you that that place in the body doesn't have cancer, or perhaps we'll be able to discover cancer in this patient way before he ever gets the disease. Great promise. Too bad that I'm almost 80 years old and I might not get to see the final results, but this really is an amazing discovery that I must emphasize again and again, all came out of the basic research that was supported by the ICRF. Thank you very much.
Well, Howard, thank you so much. Especially under these extraordinary circumstances. Phenomenal and very interesting. And of course, dear to my heart as a basic scientist, to learn how these things become translated in real life. And I know that's of interest to other people who are on the call now. Now, some folks have actually sent me specific questions, and I'm going to relate those. But if it's burdensome for any of you, simply send me a note. I have a question, and I'll sort of call you out. The first question I saw, which unfortunately disappeared because I had a break in the internet, was from Steve. And the gist of it was, what causes methylation?
Yeah, wow, that's a good, really basic question. We've actually been spending the last 10, 15 years in trying to understand how the body knows where to put these methyl groups, this methylation. And it's complicated, but we're getting to the bottom of it. And it's really a good question, because once we understand that, then we'll be able to, I hope, prevent the appearance of tumors.
Mm-hmm. Now, another question from Jerry Silber was, do you have some estimate as to how far away, what duration of time it is before such a test might become available?
Okay, so I'm very optimistic about this. It looks like within the next year or so, or 2 years, there'll be a simple test. And in another, I would say, 4 or 5 years, we'll have a test that's is more, it's deeper, and will be more sensitive. The whole idea of this test is to catch the smallest amounts, so you can catch the tumors before they do any harm. And I'm sure you all know, you've been told this over and over and over again, that one of the most important aspects of cancer is to catch it early. And this will do it. Right? Might not help in every case, but it's going to help in a lot.
That really resonates with me as well. Of course, you mentioned the idea of sensitivity, but every diagnostic test has both sensitivity and specificity as a key determinant of its value. Do you have any sense of what the specificity of such tests is likely to be, and what the risk of overdiagnosis might be?
Yeah, so I can only tell you about the specificity of determining what place in the body it comes from. And that's very high. That's higher than 95% of specificity.
Okay.
And it doesn't look to us that there's much overestimation, but we'll need to have more samples in order to really know that. I guess the idea would be that once we have the initial test, we might be able to do more sensitive tests to really see if that's the case. Once you know what you're looking for, it becomes easier.
Well, I guess there's one more question here that we could have time to take, and that is, would this test work for blood cancers as well as solid cancers, such an approach?
Wow, that's a great question. Probably less, because the bloodstream is filled with DNA from blood cells, and it's a very big background, which we're trying to solve now. But currently, that's going to be the hardest one. By the way, this test that was done, that I showed you the data for, the results, right, included 12 different types of tumors, and was able to pick up 12 different types of tumors. So it's quite wide.
Mm-hmm. It's amazing. Well, Howard, thank you so much, and thanks to all the folks who asked questions. I think we really owe you a great debt of thanks, and we're so pleased that ICRF has had the opportunity to support your research and some of this research in particular. And many, many thanks to all of you who are attending. Thank you for your continued interest in ICRF, and many of you for your strong and long-standing support. You can imagine, all of you who are on this call, that I'm a firm believer in the power of science and in the amazing success that ICRF has had in vetting research proposals from outstanding investigators and then prioritizing them for funding that many of you have provided. And so many, many thanks to those on this call whose generosity helps to foster the work of these outstanding scientists from Israel. So thank you very much. And we appreciate your taking time out of your busy day to join us and learn a little bit about the science that we're supporting. And with that, happy Hanukkah and so long.